Okay. Hello everyone. Welcome to our group 7. We will present about advantage and risk of using biotechnology in pest control. Member Gagas and Pranata with student number 138 and Rizal Zaka Nurhakim with student number 143 and then Ade Novian Wibowo with student number 145 and then Clarisa Sheiladiva with student number 178 and then Yehezkiel Aldi with student number 198 and then Rumampuk Yunita with student number 199.
The first is biotechnology related to management of plant pest organism. Definition of biotechnology is biotechnology in the field of plant protection include the use and or change of genetic characteristic of virus, microorganism, both plant and animal cell part and their metabolic product to control plant damage caused by biotic and abiotic factor. It will increase plant resistance to pathogen. Biotechnology has the opportunity to overcome difficult or unsolvable problems through conventional means. For example, at the variety assembly stage, namely in first gen identification and diversity studies, can be done with DNA microarray and molecular mark. In addition to identifying useful gene variation, can be done by allele meaning. And the second is accelerating the assembly of varieties. Can be done by haploidization and the use of mass for selection. And the third, the need for certain traits can be done by genetic engineering or through the use of in vitro culture, such as in vitro selection and protoplasm fusion.
The application of biotechnology in agriculture raises the pros and cons. According to the pro side, the advantages of genetically modified food include increasing efficiency and productivity, economic value of the product, improving nutrition, palatability value, and increasing product shelf life. Transgenic plants that are resistant to insects, herbicides, and tolerant to the environment directly play a role in increasing productivity and also minimizing the use of pesticides, which in general can have many negative impacts on plants and the growing environment itself. According to the contra side, no one knows completely what happens to the final product when you attach a new gene and then consume the mutated product for generations. The only thing that is guaranteed is that it will create surprising side effects. According to the IDEP Foundation, GMOs cause various negative impacts in various aspects of life, including A. Production costs are higher than yields. B. Increased use of chemicals. C. Loss of local varieties. D. Cause genetic pollution. And the last one is treatment is complicated.
The advantage and the role of biotechnology in the field of plant protection. The advantage, the first one is the use or change of genetic characteristic of virus, microorganism, both plant and animal cells. Second one is call part and their metabolic product to control plant damage caused by biotic and abiotic factor. The last one is in the field or of biological control. It is necessary to increase research on antagonist vegetable pesticide and increase plant resistant to pathogen include biotechnology is need to engineer biotechnology control again. And the role is the first one is engineering natural enemies of pests to become more effective again in biotechnological control. Second one is plant engineering related to gain that protect plant from intruders. The last one is engineering natural enemies with gain that determine resistance to pesticide.
Okay, and then this advances and the risk of biotechnology in the field of plant protection. So the disadvantage of biotechnology in agriculture, there are concerns that genetically modified crop may transfer genetic material into natural unmodified plant. For instance, a crop that is herbicide resistant may transfer some of its trait to a wheat, which would result in a herbicide resistant wheat. Another concern about the agriculture biotechnology center around the uncertainty of genetically modified crops, long-term biological viability. This is there is first biological pollution. There is an explosion in the population of GMO plants that beat the local plant population, and then increase herbicide use based on uh, 800 8,000 increase herbicide use based on 8.000 tuh apa sumpah aku lupa 8.000
8,000.
8,000 ya. Oh, sorry. Okay. Increase herbicide use based on 8,100 university in USA. The production of transgenic soybean use 2.5 times more herbicide than usual. And then super wheat, the continuous use of herbicide result in the growth of herbicide resistant wheat species. Okay, so and then there is allergic GMO product have never been measured for their allergy potential. And then antibiotic resistant, the use of antibiotic resistant genes in plant increase the risk of disease resistant to antibiotic. And then herbicide residue. Herbicide tolerant plant increase residue in food, drinking water, and the environment.
Pest management with biotechnology. And the example is cotton BT. Improvement of plant property can be carried out through genetic modification either by conventional plant breeding or with biotechnology, in particular genetic engineering technology. One example of a genetically engineered pest-resistant plant is the BT cotton plant. The BT gene is an isolate gene, the soil bacterium Bacillus thuringiensis. The strain of Bacillus thuringiensis or BT bacteria produce more than 200 different BT toxin, and each is harmful to a different type of insect. Cultivating BT cotton can produce various benefits for farmers. Weed management with biotechnology. Example plants label Roundup Ready consisting of soybean wool resistant to the herbicide Roundup. Two methods can be used to develop crop resistant to herbicide. The first is conventional plant breeding, utilizing lines that are known to be tolerant to specific herbicide is one approach that could confer resistance to susceptible crops from closely related species. Two, mutagenesis and selection in physiologically inhibitory concentration of herbicide, or also referred to as brute force selection, or genetic transformation of already existing crop cultivar within then confer resistance to herbicide.
Disease management with biotechnology. The implementation of molecular biology-based biotechnology is very helpful control of Candra disease based on biological, ecological, and disease epidemiology. Advances in molecular biology have given birth to various techniques that can be used in the management of Candra disease. The first is diagnosis of Candra disease. The second is early detection of virus infection. The third is identification and characterization of virus strains and leafhopper biotype screens. The fourth is monitoring of green leafhopper resistance to a varieties and emergence of virus strains and green leafhopper biotypes novel. The fifth is characterization of varietal resistance. to virus and leafhopper green. And the sixth is assembly of resistance varieties based on the resistance to viruses tangro and green leafhopper, and variety assembly tangro virus resistant transgenic.
Okay, thank you everyone for your attention, and we hope that you get a lot of insight from our presentation.